Very nice. Good morning, everybody. Nice to have you here on the Phenomics webinar series with our next session. We are very happy to have Christophe Salon with us today, who will provide us with a short summary as well on the video he provided on our homepage. And then obviously the session is very much about having your questions answered and seeing what you still want to know and what information you want to get from Christophe. So we're very much looking forward to that one. A very short introduction maybe to what's going to happen today and how we do it. This is part of the Phenomics webinar series that we have started in 2020. And due to, well, the feedback we got from you, we planned and organized a kind of a relaunch of this webinar series, also addressing the limitations we all live with nowadays due to the pandemic situation. So the new format, obviously, you know, has kind of a pre-recorded session that you can make use of. And then we have the sessions like today with a Q&A, where we can provide you with some further information based on the video you've seen. The whole webinar series is organized by the International Plant Phenotyping Network, by the Research Infrastructure Emphasis, and by the European project EPPPN 2020. Me being Sven Fahner, so I'm based at the Institute of Plant Sciences at Research Center in Jülich, mainly responsible for strategy and coordination of the European Research Infrastructure Emphasis. So what we're going to do today, I'll just provide with a very short, let's say, housekeeping rules, and then we will, I will open the floor to Christophe, provide some background information on what he's doing on his career. Then he will give a very short summary of what you've seen already in the video, and then we will open the floor for getting a well, broader discussion and getting your questions answered. In terms of how we do it organizational-wise, so if you've got any comments, questions, information you want, please put it into the chat. Throughout the whole session, you can do it now, you can do it during the summary of Christophe, you can do it afterwards, and I will pick it up and then moderate it through. While doing so, please, obviously your mics are muted and your videos, but when I moderate the questions and there may be some further discussion demand, I can open your videos as well, so that you, and your audio, so that you can speak up and well, present yourself and the question and your information demand. Right, so let's move on to Christophe Salon, who is here with us today. Nice to have you here, Christophe. Great to have you in this round. Some background information on what I know about you being a research manager or in and with INRAE, deputy manager of the large research unit agro-ecology, founder and scientific manager of the high throughput facility for PMI in Dijon. So far having been involved in, well, a large number of European research projects in different roles, also in coordination roles, associate editor of 2 journals, Legume Sciences and Plant Phenomics, and with a long list of publications in peer-reviewed journals. So, Christophe, do you maybe want to provide us a little bit of information how you get to the role that you have by now? It seems to be a quite rich career pathway, isn't it? So how did you get there?
I began starting to my PhD, or well, I can. In fact, at the origin, I'm a physiologist. I'm a physiologist, and I was trained in biochemistry and physiology in Bordeaux. Then I quit research to found my own company, which was a computer scientist company, which is still running. It's in Paris right now. And then after 5 years, I decided to go back to biological research. And so I went to Canada to work on cyanobacteria, on carbon fluxes, because my first background was modelization in physiology. And then I moved on in INRAE. It was 25 years ago. And how I ended up in phenotyping, it's because when I first arrived in 1996 in INRAE, I conceived a platform to measure isotopic flux on the plants using C13, N15, 34 sulfurous. So I had some skills in, working with, by the name in English, it's called bricolage in French, doing plumbery, everything you want at home or at work. And in 2006, my colleague said, okay, you have mounted a platform for isotopic flux. Maybe you could conceive a platform for high throughput phenotyping. So it was a large, a big challenge. And with one of my best colleagues, which is called Christian Jeudi, we took the challenge, and this is how we set up this platform. And in 2010 or 2011, discussing with François Tardieu, which is well known, he said, "Oh, you have a platform. We could set up a big project which is called Phenotyping, and what would you like to do within?" Then I said, "Well, I would really like to phenotype roots because I think it's not so much considered by the partners. They are all working on shoots, so maybe there is something to do on roots." And this is how we, with an industrial partners, we decided to work on roots. And now it's 10 years from now. We are well equipped. And recently, I got a big grant to enlarge the phenotyping facility with robots and so on. And I see that there is NAIRO on the partner. So I'm very interested because I want to collaborate with them. I had the president of NAIRO who came last year in Dijon, and we discussed together. And in a European project I'm now mounting, I think with Rick van de Zed to involve NAIRO. So it's a good opportunity to have a talk with them.
Very good. So we have one further topic to discuss then afterwards, after your short summary as well. This will get really productive. I'm very positive for today. Maybe, Christophe, I'll just put on some slides that you could lead us through in terms of summarizing a little bit more your very intense and very rich video session you had and we had on our homepage. So providing us with a kind of a very short reminder, and at the same time, providing potentially, if the people being new here in that round, with some background information so that they, can contribute to the I'll just go through the. I'll just wait for you, Christophe, until you tell me to move forward with the slides.
Okay. Well, this is just the introducing slide. In this slide, you can see one of the high resolution cabin for imaging roots, and you can see a P root, and I can tell you that on this special cabin we can have 7 microns of resolution for the images. Now you can move to the next slide, please. Well, in my talk that I think people saw, I was talking about the context of agro-ecology, because you know that well, my unit is called agro-ecology since 12 years. So it's not very original, but right now agroecology is a big challenge for everybody, and we need to reconcile agronomy and ecology, and this is why we are developing a cropping system which rely on more intense interactions between plants and plants, and between plants and microorganisms. And this is why, again, we developed the rhizotubes and the way to image the roots of the plants. And why did we do that? We did that because, as I said earlier, most of the people were working on shoots, but not on roots, because roots are very difficult to phenotype, and they are plastic in relation with the biotic and the abiotic environment. So it's a challenge. It was a challenge to phenotype roots, but we need to find a good root fence to increase the performance of the plants and to make them less dependent on fertilizer, and to help them also to have a better health. So this is why we developed a variety of tools that I presented in the talk. Tools are associated with methods, and of course, in order to tackle all the research objectives we had in the various projects, we need to gather a variety of disciplines. So for example, in this slide, you can see that we are working closely with genetic and genomics. We are, of course, working with phenotyping. But the big thing to link them is modeling. And we could do modeling at the level of the plant, which is quite mechanistic, or more integrative modeling. So you can move to the next slide, and I will show the facility. So this is the high throughput plant phenotyping platform, which is in Dijon. We have 6 different environments, and the plants are automatically conveyed from these greenhouses to the phenotyping cabin. We have around 2,000 pots, and we have 5 cabins in a near infrared, visible light, fluorescence. And we can turn the plants, and for example, for shoots, we can extract the leaves, the architecture of shoots. We have also 1,200 rhizotubes, which are automatically filled with nutrient solution and moved also using the same design. And we have 2 imaging cabins. The first one, which has 7 micron resolution, which can work for around 100 rhizotubes per day, because you have to manually import them. And you can see, for example, images from a legume plant with or without nitrogen, which means with or without legumes. Here is some focus on a pea plant, and you will see nice pink, healthy nodules at the end, also mycorrhizae. And we have also a high throughput phenotyping cabin. You can see it with a robot, which opens the shell. The shell is to prevent light reaching the roots, and you will see it's in real time. So you have blue, I think it's green after, and yeah, and then red light, and we can have images with a 42 microns resolution. And so in less than 30 seconds, you're able to image the rhizotubes. So I said it's 1,200 rhizotubes because these are the tools we have, but technically you could go up to 2,400 rhizotubes per day image. Thank you. And the next slide, please. Oh, maybe I have to tick again. Yeah, and in my talk, so I drafted some kind of conceptual work where you can see that we are considering plants and microbes interactions, and there are various research which are done on the seed quality, on remobilization of the nutrients from the soil to the plant. We have also the effect of the plant, the genotype of the plant on the structuration and diversity of the microbes in the soil. And in reverse, you have the feedback loop effect of the microbes to the plants. It can be through geochemical mineral cycles, but also for grain legumes, for example, for the symbiosis between a grain legume plant and the rhizobia, which provides nitrogen to the plant. So this is why we need to use multidisciplinary, because you have ecophysiology, genetics, genomics, agronomy downstream, and of course phenotyping, which help us a lot to have the morphometry of this compartment, but also the functions of these various compartments. Next slide, please. And here you can see a range of projects. I won't go through because I think I reminded the talk I was talking about some of them, but you have some projects we are running or we were running with. So they can, we worked on nitrogen use efficiency. It was the main point, but we worked also on interaction between plant and microorganisms, like in ANR project or EPPN. We did a recent TNA project with ILVO in Belgium. On drought stress, you can see all the project. Heat stress, biotic stresses, association between a plant, for example, legumes and cereal, and also beyond working only on nitrogen, we are working more and more on ionomics. It means we are working on sulfur, iron, it can be molybdenum. We consider at least 20 different micro and micro nutrients. And lastly, at the upper right corner, you can see some other project, of course, Phenom, which financed all the rhizotubes and the rhizocab, but also partnership with various industrial colleagues, partners like Le Groupe Roulier. We have collaboration with Le Sieur, with Limagrin, with RAGT, and so on. Next slide, please. Well, it was some question I wanted to address to the audience if we want to discuss of it or if you have some questions. For example, in the light of what I showed in the talk, what would be the bottlenecks that you are encountering in your studies? What is the main thing that you would like to do, which needs new methods or new techniques? And how can high throughput phenotyping focused on roots help you, and for which topic? And also, it's redundant, but what do you think needs to be developed? And as an example, for example, we are working on functional phenotyping, because structural phenotyping is rather easy because you can phenotype the forms and structures and so on. But if you want to assess the functions of the roots of the shoots and so on, and of the microbes and of the interactions, you need to have functional phenotyping. And we are working on this right now, trying to characterize, for example, the trophic interaction between plant and microorganisms. I'm thinking of exudates, for example, the effect of the plant on the soil. For example, how a root will affect the physico-chemical characteristic on the soil. It can be pH, structuration, and so on. Well, thank you very much. Next slide, please. This is an animation. Oh, you have to tick again. Any questions? That's it.
Very nice, Christophe. Great. Thanks for a summary on that one. Maybe I just move back one slide and just leave it there for a second. So if somebody has some direct input from the audience, just put it in the chat and then I can pick it up from there. Otherwise, maybe just let me start with one question directly to you, Christophe. I mean, these are fascinating facilities and technologies you have there. How, quite in practical terms, what would I need to do if I want to use your facilities and make some experiments? So how would I need to approach you, or what would I need to do if I'm just interested in doing my research using your facility?
Well, you have many ways. First, you can approach me directly, and this happens quite often. So you can say, Christophe, I would like to do this and this and this. So we will have a dialogue together. I will try to understand better your objectives, the kind of plants you work on, because, for example, we cannot work with, despite the fact that we are working with a large range of species. Right now, we are at more than 20 different species. There are species that we cannot work on. For example, if you come and ask me to work on beetroot, I will say, okay, I'm sorry, but you have to move. You have to move. So you can approach me directly, and I will discuss with you your protocol, when you want to do the experiment, what kind of collaboration you want. And on this point, I prefer to have a collaboration than I don't know the translation in English. It's called prestation service, which means you pay me and I will do the experiment and I will give you the results. I'm not really interested in this because there is not so much dialogue and so much interaction. So usually I privilege mostly scientific collaborations. So this is the first way. After we discuss when you want to do the experiment, and this can be the bottleneck because we have a large range of experiments, so the schedule is pretty filled. So for now, we have no room, at least for I would say 1 year. It might be quicker because we are working on robotization to accelerate upstream and downstream tools. Okay, so this is the first way. The second way is a straightforward one, except that the project will be finished this year. It was to go through a EPPN website. We are part of European Plant Phenomic Network. There is a nice range of colleagues we are working with since maybe 10 years, because before EPPN 2020, there was EPPN. So it's a smart project because it allows partners which have no facilities to apply for doing their experiment on a foreign platform, such as for PMI or PhenoArch in Dijon, or FAC in Clermont-Ferrand in France, or in Jülich or whatsoever. And the partners are paid by the European Commission, so everything is covered. the travel, subsidences, the experiment, the prices of the experiment are paid to the platform by the EU. So it makes a nice interaction and it allows people who have no real access and no real finance to work on phenotyping. And then after they can go through and do another project and precise their needs. And concerning our platform, because again, we were pretty filled with various projects and experiments, we had one big TNA project. It was with ILVO. It was called WinLeg, and we worked on trifolium and with grain legumes plants. And it was a long one because it was close to 2 months of experiment, but it ran well, and we finished it 2 or 3 months ago. So these are the main 2 ways to work with us.
Yeah. Okay. Great, Christophe. Thanks for providing that information. So I will now pick up some of the questions we got in advance to the session today and bring them forward. Focusing now on the greenhouse and the technologies you have in the greenhouse. The first one asking what would be, from your perspective, be advantages and disadvantages of the tube imaging system you use in comparison to other systems, for example, rhizo boxes? Okay, so the advantage of, rhizobox are working with the natural soil, if I remind well. So it's a big advantage because, in particular, if you consider plant and microorganism interactions, you're in the real conditions. You have the real substrate, so it's a nice setup. The disadvantage, and don't take it bad, of rhizobox is that you cannot see 100% of the roots. At the beginning, you can't see them because it works. If I remind you, you split or you put your rhizotrons like this, and the roots by gravimetry, by gravitropism, will touch the screen. So you can see the roots, but you still have some shaded roots. So it's one of the disadvantages, I mean, but it has rhizobox have lots of advantage because it's real soil and so on. I don't know the throughput. I think if my memory is good, there is around 600 rhizobox available right now. It might be the good number. If I talk about rhizotubes, and again, there is no, I'm not saying that rhizotubes are better or worse than rhizobox. It's complementary design, like you could find in Root Fair in Belgium, in aeroponics, the design from Xavier Drey, where you can see in 3D the plants without soil. So it's another design, and it's another question. So the advantage of the rhizotubes is that you can work with any substrate. We can work with artificial substrate, but we can work with soil too. And the advantage is that we can see 100% of the roots because thanks to the special membrane, we separate on the background the soil from the root. So you see 100% of the roots, and when you want to harvest them, which is done at the end of the experiment, it takes, I don't know, maybe 10 to 15 seconds to take off the roots from the rhizotubes, and then you can put them in liquid nitrogen and do whatever you want, like for transcriptomic, metabolomics, or so the plant is not sensing, well, it's sensing for 15 seconds, but you have no stress. But if you were working with a classical rhizotron, you would have to take off your roots, to wash it, to make it substrate free. So if I'm a plant, I'm subjected to drought, then you give me a nice shower, all my genes, oh, it's nice, I have water again, so all the genes could be up. And so this is one advantage. And the last advantage is that, as I said, you are free of substrate. So you have no need to wash, it's clean. You put it in paper bag or in liquid nitrogen again, and it's very quick. And this is, for example, what we did in many projects, but the last project we did was the NRA, which is called Mirga, which I coordinated on maize, and it worked pretty fine, pretty nicely. Sorry. Yeah, great.
But now, other system, you could consider the root system from Tony Pridmore, which X-ray, you have many systems to phenotype the roots. And I think every system is specific, of course, has some disadvantage, like the resolution. I was not talking about the resolution or the definition. For Rhizobox, I don't know, but for us, we can go up to 7 microns. It means you can see the hyphae. You can see, of course, the nodules, because with 42 microns, you see them pretty nicely. But we can go to very, very thin resolution, and even root hairs we can see.
Right. Digging a little bit into the topic of heterogeneity you addressed in your video. Asking very specific here, one of the questions we got: how many replicates do you need for your level of heterogeneity you have?
Oh, I would say the minimum is 3, of course, but we try to have as much as replicates as we can. So we did experiment with 12 replicates, for example, but it depends upon the design of your experiment. If you're coming and you say, I want to work with 1 genotype and 2 treatments, I can give you 600 replicates if you want. There's no problem for me. And we didn't go to a so large number of replicates, because usually people want to work with a large number of genotypes. So the minimum we are working with is 3 replicates. And what we can do in the rhizotubes, we can put 2 plants or 3 plants. So it makes no real replicates, but it makes, if you have 3 rhizotubes, it gives you 9 plants or 3 biological replicates. But the more the replicates, the more precise will be the experiment, and the easier will be the statistical analysis. But it depends on your question. If you're doing QTL or GWAS, it's going to be a different number of genotypes, so different number of replicates, because you have a fixed number of rhizotubes.
Yeah, yeah. A further one, do you randomize? And if so, how often?
No, well, it was a big question we had with PhenomysWebinars in Montpellier, because we are close collaborators. And at the beginning of the 4PMI platform, we were, like of dreams or phantasm. And we said, okay, we will randomize every day. We will turn the rhizotubes. It's going to be fun. So we'll have no hotspots and so on. But when you look at technical problems, and when you look at the time it takes, it's quite impossible to randomize all the days. So what we could do is to randomize by quarter by blocks or by line. But again, it takes a long time, because you have to remind that during the experiment, you are bringing your plants to the phenotyping cabins, but you have also to water them. You have also to give them nutrients. And according to the protocol, you might have 3 or 4 times per day to feed them with nutrient solution. So it takes time, it makes movement. So we ended up in the other solution, which is not randomizing, or randomizing at the minimum, and in fact taking into account the heterogeneity in the greenhouse. And this is why in the greenhouse, we are working again closely with INRA Montpellier and with INRA Lusignan, and we have put a lot of sensors for temperature, hygrometry, and light. And so we try to record the microclimate. And right now we are working like PhenomysWebinars is doing with a fish eye, the characterization of the climate, and try to reconstruct for each plant the climate it had during the experiment. So we take into account the heterogeneity, and we use it, in fact, for our response curve. It's a way to avoid doing randomization and spending time and money.
Right.
Big deal. Measurement of the climate is very important. I can tell you that in EPPN, we had many discussions, because we saw that some platform did not measure so well. It's not a critic again, but platform were not well equipped to measure all the environmental parameters. And when you know that the phenotype is the expression of the genome as a function of the environment, if you're not characterizing the environment, well, you're dead.
Yeah, right. In practical terms, now balancing the need for now again from the perspective of heterogeneity in the greenhouse. Now, on the one side, balancing the need for more data and for more, for more sensors. And on the other hand, the very practical questions of budget and costs. So is there any personal experience advice from your side, how you would say how to address this issue?
Could you take it again?
I think, as I understand, the question is that, you know, from a scientific perspective, you would like to have obviously a very intensive set of sensors and monitoring to observe the status of heterogeneity you have, for example, regarding light or temperature. And on the other hand, there is, in most cases, kind of a budget limitation of what you could afford in terms of equipping your greenhouse with sensors and with data acquisition technologies. So the question being now, based on your huge experience, so how would you address this kind of issue?
Well, the cost of the sensors is decreasing first. So, and again, if you characterize the main parameters, I'm thinking about light and temperature, it does not cost so much. Hygrometry is not moving, is not changing a lot. And if you're between 65 and 60% of hygrometry, which is the range, you don't really care. Except if you're doing special treatments for drought or things like this, but you don't really care. The big thing is light that you can tackle through a fisheye and through, as I said, reconstruction of the light reaching plants through the year, using the techniques which have been developed by Llorens Cabrera and Nicolas Brichet in Montpellier. And the other is to have some sensors, but it's not so costly, even to have sensors. So we have sensors, we have lots of sensors. I can tell you at the beginning, we had a thermo hydro buttons, you know, it's small chips. We had 200 of them. It was a mess because you have to configure them, you have to extract the data. It takes much more time than doing the experiment in time. And it's kind of tricky. So we ended up with sensors on blocks for a greenhouse of about 80 square meters. We have 24 sensors, which is quite sufficient. And it's not so expensive because if you consider the price of the platform, the sensors, it's nothing. So I don't know if I answered well to your question.
I think so. Yes, but I very much also understand the argument in terms of investment costs that you have for these kind of facilities, and then the kind of additional, in comparison, very small additional costs in terms of sensoring.
Yeah, you're not. I would say that to have sensors for a greenhouse of 80 square meters, I don't know the exact number, but it can be, I don't know, maybe 10,000 €. If you consider the price of a platform, it's 100 times more.
Yeah, that's right. Absolutely, that's right. Moving a little bit towards experiments and concrete experiments. Now, there's been one question asking, did you investigate below ground plant-plant interactions, for example, legumes and non-legume crops? So
yes, it is well, I'm specialized in grain legumes since 25 years. It's my favorite plant for many reasons, because it's a complex model and it's very rich. But of course, we know that if we want to introduce grain legumes in cropping system, if we want to promote them, we have to show their advantage. So we can introduce grain legumes as a sole crop, as a pure crop in cropping system, but we can also do intercropping. And if you take, for example, into account the intercropping or the association between cereals and legumes, you have many advantages because you have competition for light and for soil nutrients, which is not at the same time. So you end up with a complementarity. The cereal will benefit from the nitrogen from the grain legume. So it's a very nice topic. But there are other topics. You could also work between a crop and an adventice, I don't know, and a weed. You could say, oh, a weed will affect a crop when it's in natural condition. You could also assess the intra-variability, the intra-specific competition. For example, this is one of the projects we did with Hélène Fréville in the frame of the Solace project. She wanted to see how various grain legumes genotypes interacted in the soil through their roots. And the bottleneck when you are working with different species in the rhizotubes, and I think this is the same in rhizobox whatsoever, is to track the to which belongs the root. I will take an example. When you have, you can see my fingers. When you have two roots going to the ground, at one time they will cross themselves and untangle and so on. So the big thing is to try to discriminate between these two roots to be able to extract the architecture. And this is a bottleneck. It can be easy if the roots are different in color, which might be the case, or in texture. In such a case, we use hybrid spaces, but it can be also tricky but when they are quite similar. And for this, we are developing, we are trying to develop tools. And I know that in Wageningen, Rick van der Zed, and I think in Jülich also, we are working on this. And in the European project I'm mounting, I want to consider this. So yes, grain legumes and cereals, it's a big challenge, and it's an agro-ecological challenge, to answer more synthetically to your question.
Absolutely. So as you did investigate these below ground plant-plant interactions, now we have been talking about legumes, crops, did you see any indication, either direct or indirect, and well, nitrogen facilitation below ground effects?
Well, in our study in the greenhouse, we can't see it so rapidly, because you have to know that the experiments are not running long. In every platform, when you work on roots, I don't know for Rhizobox again, but you cannot go farther than 2 months, because again, you will have technical problems like roots will be entangled. But we see some facilitation for grain legumes and for cereals. For example, if you put in association cereal and grain legume, you will see that the cereal will grow better with less nitrogen than if it was in pure. For example, if you put a cereal in pure, you have to add nitrogen, okay? If you put a grain legume in pure, don't put nitrogen, or just at the beginning, because otherwise you will have no nodulation if you put nitrogen. This was in the film. I don't. Okay, so you have to be smart on this. The advantage is to have a grain legume which set up pretty early its symbiotic nitrogen fixation in order to take the atmospheric nitrogen, and then through a transfer can give it to the other crop. And this, we did it, and I think there is a publication from Fabien Le Souffleur. I could provide to the audience and to use the publication. We did that a long time ago. It was 10 years ago. It was not in Phenotyping platform. It was in the isotopic platform. In fact, we gave 15 nitrogen, 15 labeled nitrogen to the plant, or 15 nitrogen to the other plant, and we looked at the transfer between the two plants, and you could see that your isotope was moving from one plant to the other plant, which is definitively the proof that you have transfer of nitrogen.
Yeah. You already addressed, Christophe, you mentioned and you talked about, the difficulty in terms of when you've got the roots and they're kind of, overlapping, and how you deal with that in terms of analysis. So I think in your presentation, you also showed the analysis software or software tools that you use. Now, this is the question here, obviously, is coming from a user of such software types, because the question is how user-friendly would be the kind of analysis software that you apply. So what is your assessment?
Well, I would tell you the analysis of images is not friendly. I had a meeting yesterday with my image analyst, and in fact, you have a lot of algorithms to design. So, for example, we design lots of algorithms to have a nice segmentation, because segmentation is key. If you have no good segmentation at the beginning of the process, you're dead, because after you will have poor images. And so you can have the best software as you want, you're dead. But even after, to track the roots and to extract the form, it's quite a challenge. So we developed our geeks, our image analysts are still developing software, but more and more we are going towards deep learning. I think I showed this also. Why? Because in such a way you will move your expertise not so much on image analysis, because you can tick on the image using a classical software. You have many software which can be used, like Smart Root and so on. You can tick on the root, you annotate them, and after, if you have a nice algorithm, so this time it's more a computer scientist job, you can go towards deep learning and analyze thousands of images. So this was a big discussion we had yesterday with my image analyst, which is specialized in image. He said, in fact, my job is moving. I was trained to do image analysis, to develop lots of algorithms to track roots and nodules through, I was talking about hybrid spaces. And now I see that I will have to work more on the computer scientist side, because the throughput is so large that the time I spend analyzing my images and doing the algorithm, another experiment comes and I'm dead. And lastly, I can tell you that I don't know for the other platform, but for us, one experiment which could last around 30 minutes, 30 days, can generate up to 20 to 100,000 images, because you have 1,200 rhizotubes, you phenotype every day. So you multiply and plus the image of the shoot, it can generate thousands of both images. So if you have not a good image analysis software very quick, you have a long queue. And this is what happened to us for a long time. But now we are accelerating because we are going. Okay. Just tell me if my answer are too long. You just cut me. You said, Christophe, shut up.
No, not at all. There's no reason to say shut up. I mean, it's very interesting and would be definitely the wrong approach now, shutting up. Quite in comparison, there are quite a lot of questions still left. So there's still some things to discuss. At the same time, I would just, like to remind the audience here, so if you've got any questions and you would like to contribute to the discussion, any information you want, just let us know by putting it in the chat, and then I can pick it up from there and have it integrated into the discussion. Christophe, as we're talking about deep learning and moving image analysis, as you put it on your slides, from classical kind of approach towards deep learning approaches. Interestingly, there was one slide in your presentation now addressing exactly that approach. And what I was not sure, maybe you can enlighten me a little bit about that one, is you skipped on that slide artificial intelligence and machine learning. So basically you put a red cross over both of these terms. So now asking as a non-computer scientist as I am, so what exactly do you mean by that? So what is the kind of approach you take there?
Oh, I was a computer scientist in a former life, but no more since 30 years. But it's Michael Lambeuf, which is in charge of this, who explained me that at the origin you had machine learning, and well, he ended up telling me that deep learning is much more efficient than machine learning. So I'm sorry, I will not give you a nice answer, because listening to the question, I don't remind exactly why, but I know that in his mind, deep learning is much more efficient than machine learning. So this is the question I will ask him to, again, so he will refresh my mind about this. I'm sorry about the answer, but.
No, that's absolutely fine. That's absolutely fine.
There is a skip in the, not a skip, but a shift in the in the rapidity maybe, or in the precision of the, well, I'm not an expert on this, I'm sorry.
No troubles about that one. It's great though that we even know the name of your expert. So I guess if there are any further questions coming up, then after they did the webinar, so there's a person that could be addressed as well. I realize you've got quite a team full of expertise, don't you, and quite a lot of expertise and experienced people working at your facility, which is really great to see. Very impressive. There's one further question that I would like to bring forward to you now, addressing nitrogen use efficiency. And this question that we got said, with your study on nitrogen use efficiency, did you try to investigate nitrogen partitioning, so saying N2 versus nitrate, with empirical methods like isotopes to validate your model in this respect?
Yes, in fact, originally, as I said, I'm a physiologist, and when I did my PhD, it was the time that I think you were not born. I was already working with isotopes. So what we did for nitrogen partitioning, we used, but it was not to high throughput. We did it with the other platform, which is the isotopic platform. And in such a case, we did lots of publication. I could send you the references, but again, using isotope, you can easily track the fate of the isotope. So if you feed grain legumes with 15 nitrogen, it comes from roots. If you feed it with 15 dinitrogen, it comes from symbiosis. But it's not easy. It's really tricky to introduce N15 nitrogen dinitrogen in the plant because it's a gas. So we developed a special equipment. And what we did, we looked at the two ways of nitrogen feeding of the plant, and we tracked the fate of the nitrogen in the various strata of the plant, in the various, so it was in the vegetative part, in the reproductive part, it was also in the seeds, and we went beyond. We looked at the metabolic pathways. So we were able to see that there was some shift from time to time on the metabolic pathways which are using nitrogen. But if you look at each strata, in fact, this is the same sequence. It depends upon the phenology of the plant. So the answer is yes, you can, if I understood well the question, is yes, with isotopes, you can work on the nitrogen partitioning, the fate, etc. On high throughput platform, you would need functionalities. And I'm not sure it's rather easy, except if you work with 15 nitrogen, but not with 15 dinitrogen because it's too complicated. But you could do that in a high throughput platform. You have just to supply in the nutrient solution your isotope, and then you can harvest from time to time some plants and follow the fate of the nitrogen. It's quite easy to do. And I have something else to say with it.
No, that's great. I'm just gonna pick up a message we just got from one of our audience. I hope I pronounced it correctly. Kazuya Goto. I hope this is right. Kazuya, do you maybe want to address and bring up your point directly to Christophe by speaking out? You should be able to activate your audio by now. Otherwise, I can read it. Kazuya, can you hear me? Can you speak out? Oh, yeah, you had your audio activated for a second. Please go along, Kazuya.
Hello.
Hi.
Hi, can you hear me?
Yes.
Yes.
Hi. So I'm Kaz from Naro, Japan, and I'm now working at Wageningen University Research. Yeah. So thank you so much. So nice presentation, and yeah, nice to meet you again. So so I am a liaison of Naro. So what area do you interested in work together with Japan, or what your expectation you have? So please comment, please. Thank you.
Okay, it's a good question. And And I think you're working with Rick.
Yes.
Yes. So I think he approached you for another topic project I'm mounting. Well, I'm interested to work with Nairo because I met the president of Nairo. I don't remember his name last year, and he saw the facility. Well, he saw at least a movie of the facility. We described it, and we know that in Japan, and because I did a webinar for GST last November or December also. I know that in Japan, you're expert in many techniques. So in techniques, in robots, and also in imaging. So I'm very interested to go beyond what we are doing right now in our platform. And for two things. First, robots for upstream and downstream tools of phenotyping. Because Kazuya, you might know that a phenotyping platform is taking images. This is a fact. But before the experiment and after the experiment, you have many things to do. You have to prepare the pots. You have to bring them from one place to another. You have to wash them before and after. You have to prepare the rhizotubes, and this needs robots. And for this, I'm trying, and this takes a lot of time. Lots of people for tasks which are not very interesting, moving a pot, filling it with substrate. Well, we know that we have a machine right now, but okay, it takes time. And what I'm interested with Nairo is to work forward on having robots for such upstream and downstream tasks, but also for imaging, for looking at new techniques for imaging of fruits. Right now, we're using visible light. We are just coming into the hyperspectral way. Okay, so it's quite difficult, hyperspectral, but we know that in Japan, you're a specialist of this, so I would really be keen on a collaboration with you in Nairo. And I think Rick might have talked to you about the possible collaboration we have. We are talking about discriminating roots.
Yeah. Okay. Yeah. Thank you so much for your comment. So this April.
And deep learning.
Sorry. Yeah. And also this April we established the Robotics Research Center in Japan. So maybe we can collaborate wide area with you and WR. Thank you.
Welcome.
Thank you, Kazuya. Christophe, I know you're a very busy person, and you told me before we started this webinar that you've got a further appointment coming up. Well, it's very close by now. So for that reason, I would suggest that we close the webinar on that point. I would like you very, very much that you took your time, that you provided this video, and that you just spent an hour here with us asking the questions that we had collected and that we got back. So thank you very much for that activity. It was a great and it was a pleasure. For our audience, I would just like to provide a short outlook with the next webinars coming up. So we have the next webinar coming up, or questions and answers session on 27th May. With Robert Juncker, University of Marburg, on digital phenotyping of individual plants and plant communities in the field. As always, you will find a video in advance on our homepage. And after that one, we have the next Q&A on 17 June with Samuel Oswald from Vito on semi-automated object detection workflows using UAV imagery. Further information you can find on our website. Obviously, you can also sign up to the newsletter if you want to, where you get some further information on the upcoming seminars. And yeah, if you liked it, just tell the community about that one, tell about the options we have here. Next to that one, I would like to thank you very much. Enjoy the rest of the day, enjoy the rest of the week. Christophe, take care, and looking forward just to stay in contact and some further activities we have.
Thank you very much for the opportunity. It was a pleasure. See you again for a beer or something, some social events one day.
Yes, looking very much forward to that one.
Bye.
Take care. Bye-bye,
Bye,
all of you.